Chapter 9, Cell Communication. The main learning objectives in this chapter is to understand the four ways of cell communication, basic signaling mechanisms. We will study internal receptors as well as receptors that are found on the cell membrane. We will learn how a signaling molecule, it is called as a ligand, can trigger a signal transduction pathway in order to elicit a response. So this response can either be a gene being turned on, a protein activated, or some kind of process that is triggered inside the cell. We will also briefly talk about how yeast and bacteria can communicate. Our focus is eukaryotes though. The four types of signaling that you can see in cells is autocrine signaling. So that's when a cell will activate itself. Auto means self, autocrine signaling. This is by direct contact. The second picture is showing signaling by direct contact. So here are two cells, they are connected by a gap junction, so they can exchange ions and they can communicate very well together. The third one is paracrine signaling, which is a signal between nearby cells. And endocrine signaling is signaling between far away cells. A good example of gap junctions, right? It's cardiac cells. And they will exchange ions like calcium and so forth. That's why all the cells in the heart will work together for the heartbeat. Paracrine signaling, a very good example is neurotransmitters. And here you can see a cell that is sending out the neurotransmission. And then there is a gap. This is called the synaptic gap. The neurotransmitter has to find a receptor in the postsynaptic cell in order to pass on a message in the neurons of your body. Here's a little animation that shows how neurotransmission happens. So cell signaling or cell communication is the ability of a cell to receive the signal, process the signal, and respond to the signal. And this is very, very important for maintaining homeostasis in a cell. And there's four kinds that's listed here. Signal transduction pathway refers to the main steps that are involved. So the signaling molecule is called as a ligand. It will find a receptor in the cell membrane, which is usually a protein. So that step of this lock and key binding, which is very, very specific, is called reception. The series of molecular events that happen in the cytoplasm of the cell, where a protein, an inactive protein will be activated and the message is relayed. It's called as a cascade. There'll be amplification of the message here. It's called transduction. And when that triggers, maybe it will activate a gene in order to express itself. It is called as the cellular response. So the cell that is responding to the signaling molecule is called as the target cell. So signal transduction pathway is nothing but a pathway that will take a message from outside the cell to inside the cell in order to trigger a response. So we'll talk about the important features in every step of the signal transduction pathway. So what are receptors? So receptors can either be found inside the cytoplasm. So it will be called as a, so you can see the cell membrane and you can see the cytoplasm. So a receptor can be found in the cytoplasm. So it is called internal receptor. If the signaling molecule is very small and if it is a hydrophobic, it is able to wiggle its way through the phospholipid bilayer, then it is an internal receptor. Most of your hormones, like progesterone, androgens, testosterone, they are all steroid hormones. So their receptors are found in the cytoplasm. Then they'll find the receptor in the cytoplasm that will trigger the signal transduction cascade. And here in this case, it's turning a gene on. Whereas most drugs in general are huge molecules and they have to find a protein or a way to enter the cell. And this is where you see the cell surface receptors. So they will transmit the signal from outside the cell to inside the cell. There are three kinds of cell surface receptors: ion-gated channels, G protein receptors, and tyrosine kinase or enzyme-linked receptor. So here's a picture of the molecules that can diffuse through the cell membrane. So here's a picture of steroid hormones. They have a very similar chemical structure to cholesterol because that's the precursor. The molecules are very small and hydrophobic, and they can diffuse across the plasma membrane. So they do not need a receptor in the cell membrane. They will just diffuse inside the cell if it is small and hydrophobic. Here is a picture of a cell surface receptor, and this is called as a gated channel, gated ion channels. And you can see in this animation, the ion, when it anchors on the receptor protein, it will create a conformational change. See, it opens up, the gates open up, and it can allow all the ions to flood inside a cell. And that will evoke the cascade and the response. So these are proteins that are embedded in the phospholipid bilayer. The second type of receptor, this is the most important. It's called as a G protein receptor. And you can see that this is a very special receptor, and this is one of the most important receptors you have to know, because 30 to 50% of our drugs will use a G protein in order to enter a cell. So this protein has a lot of transmembrane alpha helixes. I think it'll go through the membrane like 7 times. It will have an external part and then a part that is facing the inside of the cell, and it will have 3 subunits. It's called as alpha, beta, and gamma subunit. And when the signaling molecule binds to the G protein, it is called G protein coupled receptor. When that union happens, GDP will be the G protein subunit will exchange GTP for GDP. It's a guanosine triphosphate becoming guanosine diphosphate. This will make the alpha subunit to dissociate from the rest of the subunits. It will activate another protein in the cell membrane called adenylyl cyclase. Adenylyl cyclase will break down ATP to cyclic AMP. Cyclic AMP is called as the second messenger that is going to trigger the signal transduction pathway. And this is how adrenaline works. It's epinephrine, another word for epinephrine. So G protein coupled receptors are very, very important because 30 to 50% of the target for most of the drugs that has been discovered is this way. We have to know this inside out. So in the cell membrane, there is this transmembrane protein called G protein. It has 3 subunits, alpha, beta, and gamma. When the ligand anchors in the receptor part, there's a conformational change in the G protein. The alpha subunit will dissociate and it will trigger the next enzyme down the pathway called adenylyl cyclase. This will convert ATP to AMP. and it's a cyclic compound. That's why the cyclase. This is called as second messenger because this is going to trigger the next step of the pathway. Amazing, right? The next protein is an enzyme. The example here is tyrosine kinase. And here, a very interesting thing happens when the signaling molecule anchors in the receptor region, the cup-like region on top. The receptor tyrosine kinase proteins will be inactive. But when they come together and form a dimer, they will be activated. And here, see, ATP is involved in it. And ATP will become ADP, but this dimerization will activate the tyrosine enzyme that will evoke other kinases. Kinases are proteins that will add a phosphate group. In other words, they will activate proteins for the signal transduction pathway. So the ligand attachment, dimerization, phosphorylation. So here it is activating the next protein in order to activate the signal transduction pathway. Another example that we see in your textbook is the epidermal growth factor. Here, the EGF is the epidermal growth factor. It will bind to the receptor called epidermal growth factor receptor. This is a EGFR is a tyrosine kinase that will work when there is a repair has to happen, when you have a cut in your skin and the repair has to happen in order to patch the cut in your skin and wound healing. This binding will trigger a cascade of reactions. You can see how like every protein is going from an inactive form to an active form because of phosphorylation. So you see that P that is being attached. And this cascade will trigger wound healing in the human body. And when this malfunctions, it can lead to skin cancer. So this is an example of TK enzyme receptors. So what are some of the other second messengers and controlling substances that can be found in a cell? One way is phosphorylation, that is addition of a phosphate group. This can wake up an inactive chemical. Second messengers, like we already saw cyclic ANP. So they are small non-protein molecules. Even calcium can be a second messenger. They can activate the signal transduction pathway. The third one is called inositol 3-phosphate. We saw this in the previous slide. So this is a lipid that is found in the plasma membrane that can convert, that can act as a second messenger. So the signaling molecule is called first messenger. These all little molecules that working inside the cytoplasm are called second messengers. So the main idea of signal transduction pathway is to evoke a response in the target cell. Or in other words, affect gene expression. So what you're seeing in red there is the messenger RNA, but you can see there's a loop. So the RNA polymerase cannot transcribe it. So what happens is all the colored ones, you can see ERK is from the previous slide where it is phosphorylated and it is active. It will activate all the transcription factors, and then that will remove this barrier, that loop that you saw. And that's why a gene can be expressed. It can be transcribed and then later translated. So we have seen a series of steps from reception, signal transduction to the response here. So how does our body regulate it? So if you want the whole thing to stop, the ligand must be removed. So these are the common ways as to how you can terminate the signal transduction. You can degrade or remove the ligand. You can dephosphorylate the phosphatases or degrade the cyclic AMP, which is the second messenger in the cell. So everything is tightly regulated. After the response happens, the whole thing will reset to neutral. If this doesn't happen, again, reason for cancer. Another very interesting phenomena that happens in our body is programmed cell death or apoptosis. Here's a very fascinating picture of a fetal mouse. So signaling transduction pathway will happen. So in the early stages of embryology and development, you can see all the toes, they are webbed. There is skin between the toes. But at a certain stage, a protein called bone morphogenetic protein is the signaling molecule that will cause programmed cell death between the toes, thereby removing the webbing. It happens in humans also, in our toes and fingers. So apoptosis is programmed cell death. How does yeast use cell communication? So yeast is a fungi. It is a eukaryote, but it is unicellular. They will usually, when they want to make more yeast, they will just, it's called budding of yeast. But sometimes it can also act like a sexual response. So there are two types of yeast. There's one cell that can make a mating factor, a chemical called A factor. The other one will make something called alpha. And when they find the receptors on the receiving cell, they will be attracted towards each other. They will grow towards each other and the nucleus will fuse. So this is a type of cell signaling that is unique to yeast. It's called the alpha and the A mating factors. So when the mating factors anchor on the receptor, it will trigger a signal transduction pathway that will make the yeast cells to grow, attract towards each other. They will form some kind of a bridge and they will come together for the nuclear fusion. Another very interesting cell communication that we see in bacteria is some bacteria can produce autoinducers. So autoinducers are signaling molecules that will be secreted by the cell, and it's actually looking for other bacteria. See, if it's one bacteria, it's very easy to treat. But bacteria are very, very smart. They use something called quorum sensing. So quorum sensing is a type of cellular communication used by bacteria that will inform them about the abundance of bacteria in the environment. So it's kind of like they will gang together and thereby they can escape the body's immune system. And also it becomes resistant to antibiotics. You know, the plaque in your teeth is a biofilm. People with, fake heart valves or like any catheters and so forth, they suffer because of biofilms because it's hard to treat because the bacteria has come together. They will produce something called pili. They will attach to surfaces and they become resistant to many of the control mechanisms. There's also an example of quorum sensing in the squid that is found in Hawaii. So it lives symbiotically in the squid and it can turn on the luciferase gene, its luminescence, and thereby helping the squid to escape predators. So these are the main points in the signal transduction pathway, and I hope that was helpful. Ask me questions.